This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on June 27th, 2024. Hello, ASV. I'm Kathy Spindler, and you are listening to the podcast all about viruses. Today, we are recording at the 43rd annual meeting of the American Society for Virology in Columbus, Ohio, hosted by The Ohio State University. Joining me today is Brianne Barker.
Hi, great to be here. For all of the aficionados out there, in Columbus, Ohio right now, it is a beautifully sunny day and 76 degrees Fahrenheit, which is in the 20s Celsius.
Our guest today is from Baylor College of Medicine, Dr. Mary Estes.
Hi.
If you like our work, please consider supporting the science shows of Microbe TV at microbe.tv/contribute. So first, Mary, we'd like to hear a little about your history. You can go back as far as you want, elementary school or college or wherever, and just tell us a little about how you got to where you are today.
Okay. So I actually was born in Virginia. I lived actually a number of different places, which I've learned is unusual for people to move around. I lived in Washington, D.C., lived in Rochester, Minnesota, lived in Tenafly, New Jersey. I went to college at a small girls' school in upstate New York, went to graduate school in North Carolina in Chapel Hill, and then went and did a postdoc at Baylor College of Medicine in Houston, Texas, and I've been there ever since. And in the midst of all that, I also did a mini sabbatical outside Paris, France, which was a great experience. So I've really enjoyed in doing that. I've learned that I love to meet people. I love to hear your stories, and to know, what you're doing. I didn't know I was going to be a virologist. When I went to college, I thought I was going to do pre-med. And I took my first biology class that we learned about immunology, which I thought was fabulous. And then I learned about microbes, which I thought were even more interesting. I volunteered in a hospital for a while, decided maybe I didn't want to be a doctor. And then I discovered, because we had someone who came back to this small girls' school who had gone on to get a PhD and learned that you could actually continue to study and answer questions, ask questions about so if you were working with a patient, it wasn't how to take care of them, but what caused this disease? What could you do to perhaps control it or learn how to do something different about it? And I decided that was something I wanted to do. So I went to graduate school at Chapel Hill.
When did you first get involved with research at Chapel Hill?
Actually, I had worked in one summer at the Mayo Clinic. When I was in college, I'd gotten one of those summer opportunities to work, and it was in a microbiology lab. We went and collected blood from chickens and purified IgE, which I'd never heard of before, but it was fun. It was a fun summer.
So tell us a little more about your doctoral work and at Baylor. Who did you do your dissertation with, and how did you get to the virus work that you're in now?
So at Chapel Hill, I started taking micro courses, and then we learned about viruses, which I thought were even more interesting than immunology at that point. And I discovered there was a virus that caused cancer, simian virus 40, small, at that point, a small DNA virus. And my PhD thesis was actually describing the proteins of the capsid of SV40. You may have heard VP1, 2, and 3, and 4. And then we discovered that there were histones that would wrap around the genome. So that was what I did for my PhD thesis. And then we moved to Texas. I got married in graduate school. My husband was a marine geologist. We had a baby in graduate school. I went with him to Texas, wasn't sure what I was going to do there, but I'd heard that Joe Pagano was my advisor at Chapel Hill. He had started the cancer center there, and he said to me there was a very good virology department at Baylor College of Medicine. And so that's where I went and did a postdoc with Janet Butel, who was the person that discovered SV40T antigen, actually. And she became the chair of the department, was a great mentor. And then I've ended up changing fields to work with GI viruses instead of SV40.
So I always found that unique that Baylor College of Medicine had a department of virology, not even microbiology, virology very specifically. It's kind of unusual.
Yeah, that was because actually there was a department of microbiology as well. And Joe Melnick, who was someone who had actually started the field of environmental virology, he had been in New York at the time that polio was coming through the country, and he was one of the first people to go out and look in the East River to see if he could find a virus. He started environmental virology. He took water from the river, found out that he could concentrate those viruses and study them. And he was recruited to Baylor. He did not want to teach medical students. And so the microbiology was there. The faculty in microbiology taught medical schools. Dr. Melnick really wanted to just do research. So he established a separate department of virology, where we just had people doing viruses. It was a great place to learn about viruses.
Cool.
So you mentioned some of your early work on SV40 and then the fact that you switched to GI viruses like rotavirus. So why did you decide to make that switch? What made you interested in GI viruses?
So I was interested actually in our last speaker telling you about all the things that he got turned down for. So when I first went on the faculty, I tried to get grants to actually work with adenoviruses. I've been working with those in the lab, and I couldn't get any money to work on adenoviruses. And I don't know how many grants I wrote. And I met David Graham, who was actually Dr. Butel's husband. He was a gastroenterologist at Baylor. And we started talking, about a lot of things. But there had been a paper published suggesting that this new GI virus called rotavirus might be associated with Crohn's disease. This is an inflammatory bowel disease that's really a big problem. And so I had all the tools to be able to look for footprints of a virus in a tissue if I could get tissues. And so one night, we were talking somewhere and we decided that, we would try and see if we get money to do that. And my first grant came from the Crohn's and Colitis Foundation, which is a good place whatever you're working on, if there's a private foundation that supports research, it's not a bad idea to look at those because sometimes they'll support young people. So we got a grant from that, and that was where we got started. It turned out rotavirus was not the cause of inflammatory bowel disease, but at that time, nobody had done any molecular work with the virus, and so I was lucky that I got in there at the ground level.
What were some of those early questions that you were working on?
Well, we didn't even know what were the proteins in the virus. So we could get clinical samples. We could grow virus in the laboratory. We could purify it. From my work with SV40, that was what I did. I could purify it. I described those proteins. So I just took that expertise, then showed what were the proteins of the rotavirus. We did gene coding assignments. We set up ways to grow the virus, how to purify it, and then it went on cloning the genes, ultimately made a virus-like particle vaccine that we then did studies with that.
I think you're jumping through a lot of years there. But so what were some of the early challenges with working on rotaviruses?
I think getting the reagents. Because there hadn't been really stocks that were available from ATCC. We got some virus from a simian rotavirus that grew in the laboratory pretty well from someone who had gotten that virus from a monkey in Africa. We discovered actually that the virus that we started growing initially was a quasi-species and it had multiple different strains. And when we cloned out individual plaques, we found that the proteins of the individual the virus grown from those individual plaques some of them had a different molecular weight, and that turned out to be a glycoprotein that could be glycosylated. There were various species of that could be glycosylated in different ways. And then I think just getting a lab started. I think the hardest thing, you don't get trained on, how do you pick the people to come work for you? I was lucky that, I ended up having great people to work for you, but convincing a first graduate student that they should come and be a graduate student with you as a new junior faculty member was challenging, but ultimately, we got great people to come work for us.
So tell us a little more about rotaviruses. Do they infect humans and other organisms, or are they different rotaviruses then for the different ones, or?
Yeah, so there are lots of rotaviruses, and there's one that really infects, I think, every species. They're in rodents and snakes as well as people. They're the main cause of actually death in young children. They cause a really clinically severe dehydrating diarrheal disease. And although we have good vaccines now, those vaccines don't work as effectively in the places where they're really needed. So there are still about 200,000 children a year that die in developing countries. And so people are now trying to figure out how to improve those vaccines. But they're in animals. They cause problems in calves and pigs. And we worked initially with animal viruses. As we moved over to work with human viruses, we found there are really differences in the animal viruses bind to sialic acid, like we've heard about influenza and other viruses. The human viruses actually bind to histoblood group antigens, which was a different kind of a glycan. And that's interesting because it turned out that ultimately the other virus I work with is called Norovirus, and that binds to histoblood group antigens, and that's a genetic susceptibility factor. If you have the right glycans on your surface, you're susceptible to infection. If you don't get infected.
So how is rotavirus infection acquired? Can it be acquired, for example, from the animals that you mentioned?
So there's not really, I'm sure there are some infections from animals, but really there's not been a lot of evidence that there's zoonotic infections. Certainly there are lots of animal rotaviruses. The recombination, you can get some genes from an animal virus coming into rotavirus, but really it looks like the most of the viruses in people are coming from getting it from other people. It's a fecal-oral transmission predominantly.
So tell us a little about how the virus causes gastroenteritis. What do we know about that? What have you learned?
So we know it can cause gastroenteritis by different mechanisms. So it grows predominantly in the enterocytes, which are cells that are on the tips of the villi in your intestine. It can kill some of those cells, and those are important in when you eat a meal. They absorb the food that you're eating, and so the virus can, if you don't have enterocytes that are functional, you'll have malabsorption. So that will cause you to then have diarrhea. We learned that the virus, actually one of its proteins, a non-structural protein, is a viral enterotoxin. That's something we found out as we were working through what do all the different proteins of the virus do. We went to make an antibody to this non-structural protein and injected a mouse with it, and the mouse got diarrhea. And we thought we must have contaminated our protein with the virus because these mice got diarrhea. And we went back a few years later, we did that experiment again, we got the same result, and this time we had injected a peptide into a mouse that got diarrhea. So we thought back, we did this before with a protein that we purified, but now we really for sure had a purified peptide. And we started to think, how else could that be causing diarrhea? And we went on to show that, in fact, this protein functions was the first viral enterotoxin that was described. And you probably all know about bacteria that cause diarrhea. Enterotoxigenic E. coli is one. There is an enterotoxin that causes it. We showed that this rotavirus protein dysregulates calcium in the cell, which ends up causing chloride secretion from cells, and that causes diarrhea. And more recently, we have found that there's another non-structural protein in the virus that actually degrades a lot of the transporters that are on the cell surface. And so if you don't have those, you also get malabsorption. And so that's a new virulence factor that was just published actually last year. Sue Crawford and other people in the laboratory have been driving that story, which was totally unexpected.
So all of that sounds terrible, and I don't want it.
Yeah, nobody wants it.
You've mentioned a little bit about the vaccine, but are we normally vaccinating here in the U.S. Most of the time?
Yeah, all children in the U.S. should be getting rotavirus vaccines.
Okay.
Yeah.
In those who are infected, can they get reinfected?
With vaccination, or the vaccine. Yeah, the vaccine is protecting against illness, not infection. Okay. You still could get infected, but the vaccine in this case was designed to prevent severe infection and death, as opposed to prevent infection. In fact, there really aren't vaccines that prevent infection. People don't know that. You think that they are, but basically they prevent disease. And every time you get reinfected presumably at lower levels, you're going to be boosted. Yeah.
But what about those who've been just infected, who hadn't received the vaccine? Can they get reinfected?
They can get reinfected, yes.
Do we know why they don't have immune memory?
Well, they do eventually get it.
Okay.
So the children, and it depends on where you live in the world as to when. Another thing that was found with studies that we did with people in India was that, so in the United States, most children get this infection at age 6 months to 2 years. So after maternal antibody that you get transplacentally wanes, but certainly by 6 months, it's probably gone in babies. And that's when the kids begin to get really susceptible. So they'll start getting infected and have disease from 6 months to 2 years of age. What was discovered was that in India and other places, lower income countries, children are actually getting infected much earlier. They may be getting infected by 2 or 3 months. And they'll get multiple infections. They ultimately will become immune, but they may have died before they have the opportunity to become immune, because they don't have access to good medical care. It can be a very contaminated environment. You can get sick very, very quickly. I can tell you, my daughter doesn't like me to tell stories about her, but she got rotavirus, and I knew what she had, but she was sick and vomiting so quickly, I had to take her to the emergency room. She was actually in kindergarten. I put her in the car and tried to get her there as fast as I could. By the time we got to the hospital, which had taken probably 30 minutes, I had to have somebody help me. She was not 6 months old. She was about 4 years old. She had gotten so weak. Someone from in the parking lot had to help me carry her into the hospital. That was really, scary. And I could see how quickly a child could die. And I knew what was happening.
Sure.
Or thought I knew.
So maybe you could tell us a little about the molecular biology of the virus and this amazing fact about the release of messenger RNA from the 5-fold axis of transcribing rotavirus particles. It's It's some interesting stuff about this.
Yeah, so rotavirus particles they have a triple layer, 3 layers of protein shells, concentric layers. The genome is a double-stranded, segmented RNA genome. And the virus has spikes, actually, on the spike protein on the surface, which we didn't know about. That was another kind of interesting story. I have a collaborator, Dr. Prashad and Wachu was at Baylor. We had purified particles and gave them some particles to look at under their cryo-electron microscope. They were just beginning to develop that technology. And when they first put these particles under the EM, they suddenly saw they didn't have any negative stain in it. They could see spikes coming off the particles that no one had ever seen before. So that spike protein binds to a receptor, which is still not well characterized. It binds to either sialic acid if it's an animal virus or one of these other glycans first. That then there's a protein receptor. There have been a number that have been reported. We're not really sure exactly what the co-receptor is. The virus gets into the cells, probably through receptor-mediated endocytosis. The outer layer comes off, and then there is a double-layered particle that stays together in the cell, and the enzymes are part of that, and the transcripts will be transcribed and come out of these holes in that double-layered particle to make messenger RNAs that go out into the cytoplasm.
And then from there, so more RNA, more proteins, and how and where does assembly take place?
So the virus is assembled. The proteins that are made will form something called a virus replication assembly structure, or it's called really a viroplasm. We don't know all of the details of how the genome is replicated, but it takes place in there. There are two proteins that are glycoproteins that get made and inserted into the endoplasmic reticulum. These viroplasms are basically adjacent to the endoplasmic reticulum. And we've recently shown that we call them lipid droplets, endoplasmic viroplasms. The virus induces lipid droplets there at the endoplasmic reticulum. It replicates the genome. There are there's a glycoprotein on the capsid. We don't know exactly how the capsid is remade, but it's made in there. It will bind to our little enterotoxin is inserted into the endoplasmic reticulum that the inner capsid binds to. The particles bud. This is a non-envelope virus, but they bud into the endoplasmic reticulum and make a transiently envelope particle, so it's very interesting. That envelope is lost, which we don't know exactly how that happens either. But in the process of that, the spike protein is also put on. Those membranes then move. The autophagy is also induced, and the membranes will then move into autophagic vesicles. And ultimately, the virus is released generally lytically. There's some evidence that there may be some particles that are released in some kind of an enveloped vesicle too, but exactly depends on what cell you're in and how that works.
So I want to make sure I heard it right. The enterotoxin then gets packaged?
So it's apparently, as far as we know at the moment from what's published, it's not actually packaged, but it serves as an intracellular receptor as well. And it's inserted into the ER. And in doing that, it's also dysregulating calcium regulation.
Okay, so once it's in the next cell and the enterotoxin is produced is where it produces the enterotoxic effects.
Well, and the enterotoxin is also itself secreted, we think, outside, and it can affect neighboring cells.
Got it.
And Joe Heiser recently has shown that the enterotoxin also can, when it's releasing calcium from the ER, does it in different ways, and it also induces ATP that's released from cells that can make waves of calcium dysregulation in adjacent cells.
Okay, so for those of you who are thinking about, to me it sounds like a really cool virus that I've never worked on, and there's a lot of things to do. What would you say are the most challenging or outstanding questions that we need to work on for rotaviruses?
So the things that the lab is now working on is to try to understand, we're using the virus on a molecular level to try to understand what it does to the cell. So these lipid droplets being induced. Lipid droplets are involved in the replication of other viruses, hepatitis C virus and so forth, but nobody really knows exactly how they're made. And we've now identified, one of the rotavirus proteins that's involved in inducing the lipid droplets. We really want to understand that in much greater detail. The virus induces, there was a talk last night from the lab, the student, the virus induces PLIN1 proteins, which are proteins that will coat lipid droplets. And we're trying to understand really the details of how that works. And how that's involved in more detail for not just general how lipid droplets are made, but also what that really is doing to help the virus replicate.
So that's very cool. I will admit that we have some suggestions or some comments from some of our other colleagues of things they wish they could ask if they were here. And one of the things that was mentioned was that you have an amazing paper on the minimal infectious dose of rotavirus in piglets that is only 1 PFU. And so I was wondering if you could talk a little bit about that and explain a little about that and maybe why that might be.
Well, it says the virus is very, very infectious. Those were amazing studies. Again, studies I did in collaboration with David Graham. Those were done in germ-free piglets that we set up the model. Linda Safe here, you heard from yesterday, works with rotavirus, and they have an unbelievable facility in Worcester where they work with germ-free animals. We didn't have that at Baylor, but we got some isolators and we set it up ourselves. And so you would have a sow that, is pregnant. You deliver the babies, you dip them in Clorox and put them in this incubator, and then you would feed them different doses of virus. You can do this in people if you have a human challenge model in people, but you give them different doses of virus, and then you calculate what's the lowest dose that you can give them to infect. And yes, in the piglets, these are germ-free, so they don't have an immune system, and they haven't been given colostrum. So they're very susceptible. And what we found was giving them the virus at just one PFU. It's probably 100 to 200 particles, but one infectious particle that we could measure could make a little piglet very sick.
Yeah, that's what I was wondering. When I hear those kinds of things, I think, oh, well, the animal is a better assay of the infectious titer maybe than a plaque assay that's giving you plaque-forming units. So particles.
It could be.
Yeah.
How does that compare if the animal does have a microbiome?
So we haven't done those studies, certainly not in the piglets. There are people that are working with microbiome, and there is some suggestion that some of the reasons that the rotavirus vaccines don't work as well in the low-income countries has to do with the microbiome of what are in children. So there is an active group, our active groups. I'm not personally working on that, looking at what are the bacteria in the intestines of those children that may inhibit the replication. There are also viruses. Many children, and particularly in low and income countries, may be infected with many viruses. They get infected with the rotavirus. They may already be infected with other viruses. And those other viruses can actually interfere with the rotavirus infection. I mean, it's like with polio, we've known this for years. It's the live polio vaccine originally didn't do well in developing countries, and probably for the same reasons. That they're interfering viruses that might be in the intestine of those children. Now we're talking about as part of the microbiome. The virome is part of that. There are these other things that are competing present in the intestine of children that when they get either the vaccine or potentially the virus, they might not always get a good take.
Are there other things that we need to know in order to perhaps make improved rotavirus vaccines?
Well, so there are a lot of people working on that, trying to get. We think that if you could get a virus to grow better, you'll get a better vaccine response. I think the vaccines are just not growing as well in the children. But there could be maternal, factors in breast milk that, particularly in situations where the babies are being vaccinated quite young, that may interfere. And there are people trying to work on improving that.
So does that mean that the current vaccine is a live attenuated vaccine?
Yes.
And what's the vaccine schedule? How old did children
So
get
it's
it?
given in the standard vaccine schedules. I'm going to not remember the exact weeks of age, and they're a little different in different places, but yeah.
So are there other vaccines in pipelines that would not be the live attenuated type that you know of?
So people are talking about that, but there have not been any clinical trials in making an inactivated vaccine. But I think the idea is, I think the world is a little bit going towards not having live attenuated vaccines. And so they're thinking about trying to make an inactivated vaccine. We have a virus-like particle candidate vaccine that with proteins that were expressed in baculovirus that will self-assemble into a virus particle and in all of the preclinical studies we've done, it's worked well. So there's a couple of inactivated vaccines that are now people are trying to develop, but they haven't been tested that I know of in clinical trials yet.
So you've mentioned this already, that you also have spent some time working on another GI virus, which is Norovirus. So a lot of people might think about a lot of similarities between Norovirus and Rotavirus, but how are they different? What are the big differences in Norovirus in terms of pathology or disease or transmission?
So Norovirus is a calicivirus. It's not a rotavirus. It is a much smaller virus, and it has a single-stranded RNA inside of it that has a VPG protein. It's more like picornaviruses in that sense. It's covalently attached to the 5' end. It's a virus that until a few years ago we couldn't grow. Norovirus infects all age groups, so it's not just predominantly in children. And it kills also about 200,000 people a year in the world. Generally, they are at the two extremes of life. So children they get very sick, and then people as you age, many people in nursing homes may get norovirus and could die for that. It is a virus that has been difficult to work with. For us to start working with it, Al Kapickian is the person, he was at the NIH. He discovered this virus. Actually, it was the first virus shown to be associated with gastroenteritis. And he discovered it by getting stool samples from people that had been sick. And he got antibodies, after people gotten sick. He mixed the two of those together and put some of that under his electron microscope, and he saw virus particles. And at that point, everybody in the world thought that the only things that caused diarrhea were parasites and in particular, and many, many bacteria. But Al showed that here were these particles, and then when he started looking at other cases and samples of diarrheal disease, he could find those particles or something that looked very like them in all of these samples. It turned out to be norovirus, and it's named after the first outbreak that was well characterized was in Norwalk, Ohio. People here may know, I don't know exactly where that is.
I do. I looked it up.
But you did? Close by?
It's about an hour and a half north of here. Not quite to Sandusky, which is on Lake Erie. So between Cleveland and Toledo.
Okay. So there's a picture of an elementary school there, which was where the first outbreak had occurred, and the CDC came in and studied that outbreak. Al Kapickian showed there were virus particles, and you couldn't grow the virus, and they ended up they did human infection studies, and they showed that a filtrate from stools from those outbreaks fed to adults made them sick, and they could prove that then there was an antibody response in those people, and they could repeat that immune electron microscopy experiment and show that those people that were sick had virus particles that were clumped by antibody after somebody had responded to that. That virus was really hard to work with. For us to get started with that, I got actually some samples from Al Kapickian, and we had to we set up David Graham again. He liked challenges, so he had set up the pig model. We set up human infection model in Houston, and we fed people, put them in the hospital, fed them some of Al Kapickian's, the virus from him. They got sick. We collected a lot of material over several years, and we didn't know what kind of virus it was at that time. We cloned the genome from the material that we'd gotten from those volunteers. That was difficult because we didn't know. Were we looking for an RNA or DNA? We eventually found that it was RNA virus, norovirus, and then we couldn't grow it, so we cloned the genome in 1990. We were able to get it to grow in our lab in 2016 in these new cultures called organoids that are they're non-transformed cultures made from biopsies of people. You go to the gastroenterologist and will take a biopsy of your intestine, and they're non-transformed cultures. We grew them up. They're called mini guts. This was technology we didn't develop, but when I read about that, we tried to grow the virus in every cell line that we could think of, and it wouldn't grow. These non-transformed cultures, though, do grow the virus, and so we're now in a new era of trying to get it.
So if I did the math right, it was 26 years of not having a cell culture system for growing these viruses.
We weren't working for it along all of those years, but
for the field.
Yes, for the field.
Sometimes you'll hear about viruses for which that you can't grow in cell culture, and you might just steer away from them. But Mary did not.
I think the thing. So I tell people. I mean, I love to read. I was thinking of Felix was saying his parents had a lot of books and he read. I mean, I've always liked to read, and so I read. I'll read almost anything. And I will read widely. And the other thing I believe in science is that technology, probably developed not in your own field but in some other field, will really make a big difference for the research that you're doing. And there were two examples in my career of that. One was with rotavirus when we wanted to express the proteins. Most of the colleagues that were expressing proteins were using bacteria. You could use yeast, but I had no idea how to work with yeast. And then I read about this system called the baculovirus expression system. And we were one of the first people to put a mammalian gene into baculoviruses, and we got lots of proteins. That's how we got our VLPs. If you expressed the rotavirus proteins, they would form this triple layered particle. I mean, it was amazing. And then the second example of reading widely and taking something from another field were these organoid cultures. And I think we were maybe one of the first, if not the first people to test them for trying to grow non-cultivatable viruses. And now they're used, you can make organoids from any epithelial cell, really. I mean, we have an organoid lab now at Baylor. We have cultures from all different segments of the intestine. We have them from the liver. We have vaginal cultures. We have bladder cultures. So you can make them from almost anywhere, and it's amazing what you can do with them. They really represent the person that they came from. So we know cultures from babies are different than cultures from older people. And the viruses grow differently in them. So there's a new system that's really interesting to use to study viruses.
So you mentioned these organoid cultures and how helpful they were to start to be able to culture the virus. Do you think that is because of a variety of cells in them, or because of the sort of physical anatomical differences, or because they're non-transformed? What about it made that successful?
I think it's a combination of things. I don't know why the virus won't grow. So the only intestinal cell lines really that we had to try to grow the virus in were all cancer lines. And we don't know the receptor for the virus yet. I'm sure, my guess is that in the cancer cell lines, either the receptor is not there or there's a signaling pathway that is in that cancer cell line that won't let the virus grow. I don't know. So I think being non-transformed is making a difference. Some of we know the virus is growing predominantly in enterocytes. And if you take normal enterocytes out of the intestine, they'll die. You can't get those cultures to stay. You can't just passage them, the enterocytes just die. So I think we have a way now where we have a source of non-transformed enterocytes that the virus will grow in. Yeah. You also mentioned a little bit about norovirus and pigs. Are there other animal reservoirs, or so for is this a zoonotic virus? Yeah. Again, there are many noroviruses in animals and people. There's no evidence yet for the noroviruses really that the animal viruses are spilling over into people. But some of that is always a problem of how much sampling are you doing. Although the viruses from people, we haven't found a virus people really that we know has come from animals in terms of its sequence. The sequences are different. Yeah.
So can you tell us a little bit about the variety of strains of norovirus? I think it's true for mouse norovirus and for human noroviruses. And I was trying to think this morning about what would be a parallel in another virus system. Is it like rhinoviruses? And because they seem to have very different phenotypes, I think. Yeah, so the human noroviruses, what we know, they're classified now into
genogroups or genotypes. The murine virus is a little different than the human one. We think the primary way that it grows is in the enterocytes. The murine viruses, we've heard, will grow in tough cells in the mouse, and some of those strains also grow in macrophages and dendritic cells. So the tropism is a little bit different. The human viruses, we have sort of two major genogroups, genogroup 1 and 2, but there are a couple other that still infect people. It's perhaps more like the rhinoviruses, but we don't yet know how many serotypes we have. So we're working with genotypes. It's just all genetic classification at this point. Now that we can grow the virus and we can do neutralization assays, we were just talking about this last week, that we really need to start to try to see if we can characterize how many actual serotypes there are of the virus. That's a question for vaccines. There is a bivalent vaccine that we actually made that's now being developed by Hillevax. There's a clinical trial going on at the moment in children. All the studies, it's been a very safe vaccine, and it's very immunogenic in adults and in different age groups. But one of the questions is, all the people that it's been given, almost everyone it's been given to up to this point, are people that have had a natural infection, so they're primed. And so one of the questions is, if you give it to a baby that's not primed, it's a primary infection or primary inoculation, is that going to induce antibody that will be broadly protective for the different strains? I think there are some epitopes that are cross-reactive, but we'll have to see. I think we'll get data by the end of this year, maybe, to see how that's working in the field. It's going to be a fun year.
What's the platform for this?
It's a virus-like particle vaccine. Yeah.
I know everyone's shocked that I have a question about the immune system. But are people able to get reinfected with either the same or a different genotype? And what kind of immunity is induced?
So the immunity is not completely understood. You can get reinfected. We were able to infect mice or inoculate mice with one strain of VLP and make monoclonal antibodies that would recognize other strains of the virus. So that's one of the reasons that I think there are broadly cross-reactive epitopes. And recently now there have been monoclonals made from humans who've been infected that have been shown to be broadly cross-reactive, and they bind to a special site on the virus particle that was thought to be occluded before, but now apparently the structure of the virus has a raised and a closed conformation, and some of the broadly reactive monoclonals are bound to, they only will bind to the virus when it's in the raised conformation. So that's a new little wrinkle that's very interesting, and we'll see how these vaccines do in inducing protection.
That's good.
So I guess I want to know the same kind of thing. What do you think is the burning question for noroviruses? Or the most challenging thing that you would like to see?
Well, so the thing we're focusing on, which I think is important, is what is the receptor? Because we're working with these organoid cultures. It's fabulous. We can grow them. You can do antiviral testing. We can measure neutralizing antibodies now. But they're complicated and certainly more expensive than just having. These cultures are multicellular. We can't do a CRISPR knockout in the cultures. These are cultures that you grow them it's a proliferating culture where the stem cells are being stimulated, and that's how we keep them going. They're good. The cultures we made in 2011 we're still using, so you can freeze them down and then pull them out of the freezer and keep working with them. But then to infect them, we take away the growth factors that are stimulating the stem cell, and that stem cell differentiates into 4 or 5 different cell types that are normally in the villus on the intestine. So you have a multicellular culture. If you tried to do a CRISPR knockout, and our virus only infects the differentiated culture, so you've got to have the enterocytes there. It doesn't replicate in the stem cells. So we're doing biochemistry to try to find the receptor. But the idea is if we can find the receptor, we'll be able to put that in a classical cell line that then everybody in this audience, if you want to work on noroviruses, you could work with. So that's one of the to me, one of the most important things. The other thing is, although we can grow the viruses in these cultures, we can't passage it indefinitely yet. So we're studying the innate response. There's a very big innate response. And I think, so the normal acute infection really only lasts when we used to feed the virus to adults. We knew when we gave them the virus, we gave them, cocktails of Norovirus on Thursday night at 6 o'clock. And if they were going to get sick by Saturday at noon, they were sick. And people would have a variety of outcomes. Some people would just get diarrhea. Some people got vomiting. The people that were really unlucky would get both. And then it's a short infection for most people. By Monday morning, they would be fine and they would go back to work. What we learned after that was that they're still probably shedding virus for a month. So the virus doesn't just get cleared immediately. So I think understanding the immunity and what really is clearing the virus is something that's important. And we're really trying to now figure out, we've had some success in knocking out, if you make a STAT1 knockout, we have one virus strain now that will grow better. But there's a pandemic strain that and then there are variants of that have been crossing the world every couple of years. They change. That virus could care less if whether there's a innate response or not, and we think it's got a way to overcome the innate response. And I think that's probably why it's the pandemic virus. So we need to understand more about that innate response to, I think, also get the virus to replicate well.
So I don't want this one either then.
Huh?
So I don't want this one either then.
No. And if you're immunocompromised, I mean, people get norovirus and there's no way to treat it or clear it. And so many people are transplant patients that are on immunosuppressive drugs, and they'll get this and they're sick basically indefinitely, which is really bad.
Back in those experiments where people would either come down with diarrhea or vomiting or both, were there some people who never got sick? And
Yes.
what's an explanation for that?
And so the explanation for that, I mentioned before that there is this genetic susceptibility or resistance. So there's a gene called fucosal 2 transferase gene that is important in putting fucose molecules on. It could be, we think it's probably a protein in your intestine that the virus binds to. We know the virus binds to these fucosylated molecules. And there's about, for the Caucasian North European population, 80% of people are secretor positive, which means you have this fucosal 2 transferase gene that's functional. But 20% of the population is negative. And what we found in our volunteer studies was that the people that didn't get infected at all, these were all given the same inoculation of virus, were secretor negative people. So they had a FUT2 gene that wasn't functional. They didn't have fucose in the intestine, and they didn't get infected. And that's been something now that when you look at epidemiological studies, you can show that across the world. Of course, we mentioned before there are many different virus strains, and of course some of the viruses use a slightly different glycan, and so there are some viruses now we found that do infect the secretor negative people. So you can't be totally resistant, but those viruses are not circulating as much, probably because there's not as many people, susceptible people around. And the other thing that's been fabulous about the organoids is that we make them from people who are secretor positive or negative. The virus grows in the secretor positive cultures. If we knock out that one gene, the virus doesn't grow in them, and it really proves that is what is important. And if you take a secretor negative line that the virus doesn't grow in, put the fucosal transferase gene back in there, the virus will now grow. And those few viruses that we know that grow in the secretor negative people will still grow in the knockout secretor positive. I mean, you take the secretor positive, knock it out. Those will still grow there. So the cultures are reflecting the genetics. They really are like mini people, or a mouse, except it's a human mini gut instead.
So one thing that was sort of interesting for us in terms of preparing is that you have had such a prolific and excellent career and have over 500 amazing papers at this point. So we can't ask you about all of them, but I want to know, what are you excited about that we didn't ask you about?
I think we hit the top, the key things. I mean, I think to me, being able to grow this virus is pretty amazing to identify the proteins of rotavirus and find an enterotoxin that wasn't recognized before. And then now more recently, we found a new virulence factor. It's been pretty fabulous. I mean, there's been great basic science with both of these viruses. And I think the one thing that I think was important that I felt I really enjoyed doing was working for viruses that really cause disease in people. And it's been challenging to work with them, but anything that somebody told me once, anything that we discovered had the potential that we might really be able to help people recover or perhaps not even get infected or get disease in the future. So I've ended up doing translational science, kind of doing it because that's where the viruses were, and before everybody was trying to do translational science, and that's been really fun to do. It's hard, but it's been also wonderful and very, it's been fun to interact. To get the things done we did, we had to interact with clinicians, and we've had a great relationship in doing that. One of the things that I did we established a graduate program at Baylor College of Medicine called Translational Biology Molecular Medicine, where the goal was to be able to treat, it could be MD PhDs, but mainly PhDs, to come into graduate school, and they would, in the first 2 years, they spent about a half a day a week, or in the second and third year, working with the clinician. So they had a basic science mentor and a clinical mentor. They would go into the hospital, work with, and they would go on rounds or do various things over those 2 years and get exposed to something related on a clinical side of what their thesis topic was, but become comfortable and learning more about how the clinical medicine works and what were the problems. I mean, the physicians are fabulous, but some of them are so busy taking care of patients. There are things they want to solve, but then if you are there talking to them and listening to them talk, you'll find there could be a simple experiment you could do to help them be able to take care of their patients better. And so that's been really fun. And the students, they've been fabulous and are going off and doing lots of different things.
Yeah, it sounds like a really great experience.
Yeah, it was good.
A lot of students would like. So I have some questions because you have a really long history at ASV. I had access to the records, surprisingly. And you gave state-of-the-art talks in 1986 and 1991, and then you have given 5 plenary lectures, all of them endowed, 1995, 2000, 2004, 2008, and 2019. A couple of those were Lois Miller lectures, which is near and dear to my heart, and Lorena's, a famous baculovirologist. But I'd like you to maybe tell us, and Mary was ASV president in 1996 to 1997. So if you could tell us a little about differences between then and now, what's similar, what's different.
I mean, there's a lot of things I think everything is good. The society was much smaller in the beginning, for sure. This is the first meeting we've had that's in a convention center. The previous ones were all at campuses. So it's a slightly different feel, but the talks have been fabulous. I loved coming in the beginning. I was teaching general virology. And as I said, I love to read, but I also just like to listen to people and hear what they're doing. So hearing the morning talks, they're not all about the virus you're studying, but there's so many interesting things. I got this button. I love virology. This morning's talks were fabulous. Actually, they've all been fabulous this week. So that's been terrific. And you meet lots of people. You hear... I tell my students to go listen to the talks, pay attention to which talks you liked and what made the difference. Why was that talk that maybe it was the data, but a lot of the times it's was the font big enough on the slide that you could see it, and little tips like that in terms of learning how to communicate with people. I don't know. I mean, all the meetings are good. I'm not always good at coming up with
Sure, fine.
precise things. I hadn't thought about that, but I could tell you tomorrow, I'm sure, a long list.
Right. Yeah. And just things in the society overall, being at all those lectures, but also being in leadership positions. I'm sure you've seen a lot of things that are similar and different from.
Yeah. I think the other thing that's good, because as the society has grown, there are more committees, more people are participating in the society. We had committees before, but they were probably smaller, and there weren't as many committees that are addressing lots of different opportunities to help people network and get together. And I think that's really all good, all great.
So you've mentioned a little bit about the program that you were part of at Baylor, and you also mentioned back at the beginning that one of the hardest parts was trying to figure out who to have in your lab. So from all of those types of experiences and everything else, what advice do you have for trainees or for new PIs?
It's still hard to know who to have in your lab. I think now there are more opportunities. When I started off, I think now faculty coming in, there are opportunities, lots of training that young faculty get that we didn't have an opportunity to participate in. So ultimately, there were, I think the Burroughs Wellcome Fund wrote a book about, how to start your laboratory that I read. I also read that, and which talks about what are things to consider when you're interviewing people that you might hire for working with you. A lot of it is interpersonal skills, and some of it you just have to kind of make your best guess after you've talked to somebody. Is it someone you really think is going to be a good person to be in the lab? The other thing I've learned is that if there are not good fits, that you need to separate yourself. But I've learned, and there have been times where we've had to have people leave the laboratory. But generally, what I found was that if you sat down and talked to somebody, it turned out they weren't happy either. But neither one of you wanted to say that this wasn't really a good fit. So, I would ultimately say I think it's not a good fit. And I'll help you find something that is a good fit if we can figure out what you want to do, but this isn't it.
Yeah, I think my experience has been that, you think about the faculty members in your department, most of them have trainees. So there's somebody for every faculty member, and there's a place for every trainee, and figuring out if a fit isn't good is wise, and that explore some other fits. And it may be not somebody that you did a rotation with or anything like that, but now with a little more time, you can reflect back and figure out, who would be good at this point for me in my career. I'm saying this having been somebody who changed advisors after 4 years in the PhD program.
Wow.
And so I'm not recommending that you wait that long, but in my case, things were good up until about 3.5 years. And luckily I found John Holland, who you heard Raul talk about him this morning, as a mentor and a great lab to work in. So finding a good fit is important. I agree.
Yeah, I had one student who ultimately got a master's degree, and she was extremely bright, but she actually really couldn't get anything to work in the laboratory. We
call that stone hands.
Yeah, well, but she went on for like 2 years, and nothing had worked. And we finally sat down and had a conversation about it. And, she wasn't someone, she was from a family, you don't quit. That was so that no matter what, if it wasn't working, you were going to figure it out. But she didn't really know how to figure it out. And we sat down and had a conversation. She went back and talked to her family, came back on a Monday with a big smile on her face and said, I'm going to get a master's degree. And she's gone on, she's become, she's writing, doing science writing, is running a big program at MD Anderson across the street. So, it was a win-win. She found what her niche was, and it wasn't to be at the laboratory bench. So I'm guessing that I might know part of the answer to this, but do you have any other advice for trainees? I'm guessing part of it might be to read widely. Exactly. So I think, read widely. I think the other thing I've been, I think, successful in my career because I've really had fabulous people to collaborate with. Prashad is a structural biologist. People know we do great stuff. We've been working together, 30 years now. He does structures. People know that I don't do structures, but we may have some new biology ideas that they'll follow up on. It's just been really fun. So I think picking your collaborators, if you can pick them, is great, and not to be afraid to work with people where you don't understand everything that they do. I think in the beginning, I told you I went to a small girls' school. To be honest, I was quite shy. I didn't say much in graduate school, but I listened a lot. That's not a bad trait. But I think people maybe thought I couldn't think about anything because I didn't say much. It took a little while to gain some confidence in that. But I've just had fabulous collaborators, and we love to talk about science, and we've been very effective in doing that. Persistence pays is the other thing that, I didn't get grants when I was trying to get, as a young faculty member, the tumor virus grants. And when I did get money from Crohn's and colitis to start with rotavirus, first grant I sent to the NIH on rotavirus, the comments were, Dr. Estes has never worked with an RNA virus in her life. What makes her think that she can do that? And I didn't ever get always of my renewals, didn't always get funded. Sometimes I was too ambitious. I have too many ideas. I've had to learn. I want to do all those ideas, but that's not what you put in your NIH grant. You pick 2 or 3 of those that you think maybe are something that someone else might be interested in. That's been a better strategy, but I still don't always get grants. I had one. I wrote one SARS grant, which with Dr. Prashad. We thought it was a great idea. We got very different. Was to do something looking at glycans, histoblood group antigens. It went through review 3 times. Frequently, we would have 2 reviewers that thought it was great, and we've had the 3rd reviewer would say they thought that this was the most ridiculous idea in the world, and it got tanked. So finally, after we just quit, so try things, but don't get discouraged. I think what is important as a young faculty member is to be in a circumstance where people will support you. It takes time to get, and I think today it takes longer than it probably did. I thought when I was younger, it took me a long time, but I think it's taking even longer now for some people to get their first R01. But, just get advice from people, get to know your program officers that really helps. They can be very helpful for you as your career goes on.
Okay, so we're getting to a point where we need to wrap things up, but I want to ask you one more question, and that is, if you've ever thought about what would you have done if you had not become a scientist?
I might have become, thought you might ask me what, if I didn't do what I'm doing now, would I do something else in virology? And I might have become a plant virologist, actually. I think there's a lot of interesting things with plants. Mm-hmm. And I like to garden, so.
Okay. Very good.
That's fair.
Sounds good.
So.
All right. Do we have any other questions, Kathy?
I don't.
All right. You can find TWiV at Apple Podcasts, Spotify, and microbe.tv/TWiV. We love to get your comments and questions. Please send them to twiv@microbe.tv and consider supporting the science shows of Microbe TV at microbe.tv/contribute. I really want to thank our guest today, who has been absolutely amazing. Mary Estes, thank you so much.
Well, thank you for inviting me.
I'd like to thank Kathy Spindler, who is a professor at the University of Michigan. Thanks, Kathy.
Thank you.
And I'm Brianne Barker from Drury University. We'd also like to thank ASV and ASM for their support of TWiV, and Ronald Jenkins for the music, and Jolene for the timestamps. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
I'd like to get your picture, so unless you're in a rush, hang out.
Oh, I didn't even think to take pictures of you guys. See? Act like you're listening intently and very excited. Oh my gosh, I have to zoom out.
Yeah, pretend we're saying really exciting things.
Wow.
See, like
pretend you're enjoying yourself.
That's a
Wow. There are a lot of you. You did well. Give yourselves a hand.